Any questions yet? Oh, actually, yeah, any questions yet about the exam real fast since you've had a chance to look at it for a minute? Common mistakes, stereochemistry and the amino acids, not showing it. Told you had to. Not realizing that if you draw them oriented the exact same way, even if you tell me, all facing down, you don't tell me the stereochemistry, I don't know what the heck that means. Proline, again, presented a slight challenge to some people. Similarly, but not most. It was actually really good. Y'all did pretty well with proline. A few of the amino acids presented other challenges. So make sure you learn those, because I can tell you right now, you'll find another question like that appearing again on the next test. Any questions? Okay, let's start with enzymes. Welcome to the world of enzymes.
Wait, do we get to keep these?
Yeah.
All right.
I know, they're collector's items. If you want, I'll sign it for you. I really don't sign things generally. It costs $5 for you to sign documents. That's usually what I charge. But for you, I'll only charge $2.
Oh, might help.
Well, no. Frame costs extra. All right, and how enzymes work. Y'all pretty comfortable handle on enzymes, right? Should we skip the whole section on enzymes and I'll just test you on it?
Might need a refresher.
Just a little refresher?
Yeah.
More than that, they do all sorts of fun stuff. So, enzyme catalysis is really the how biochemistry got started was looking at enzymes and discovering that this catalytic activity of these biomolecules. So frequently the interaction with proteins, we talked about proteins and ligands earlier, last section. It's usually followed by some covalent changes in the ligand. And if this is the case, then it's referred to as an enzyme. The protein is a catalyst for the chemical reaction, is unaltered once the reaction is complete. By definition, a catalyst is a compound that can accelerate a rate of reaction, and in the end is unchanged. It does not mean that it doesn't get in the process does not get transformed, but by the end of the process, it is restored, just original form. Okay, and we'll see this actually happens in the enzyme catalyst. In such cases, the protein is known as an enzyme, and the ligands are referred to as substrates. In some cases, the catalyzed reaction may be coupled to other processes. You have mechanical movement of work, molecular transport, other chemical reactions, et cetera, et cetera, et cetera. And we'll go through some examples of these, and then we're going to come back and revisit these again as we go back and talk about various events. We start talking about enzyme catalysis originally, then we start talking about how synthetases work in enzyme catalysis, and we relate that to ultimately we talk about protein biosynthesis and degradation and post-translational modification. We'll be revisiting some of these things and we give some other new additional examples of enzyme activity in these processes. Enzyme catalysis has been studied extensively throughout the 20th century and continues to be an area of considerable interest because many of the drug targets we're interested in targeting for disease, especially whether it's a metabolic disorder, whether it's a pathogen-derived like bacterial pathogen or viral pathogen, oftentimes we're looking at affecting the enzymes that are critical to the life cycle, the disease cycles of the organisms, right? Anyone who took 465 Biochem Lab, they did homoserine transuccinylase, which is a key enzyme in the metabolic biosynthetic pathway of methionine. It's the first key step in that process, the gateway step to that process for the biosynthesis of methionine in bacteria. And it's different from what we have. It's a unique enzyme, and therefore it's of interest as a potential drug target because we can hopefully theoretically inhibit it while not as it negatively impacting our own biosynthesis of methionine. And since methionine figures so prominently in protein biosynthesis, protein translation, the first amino acid, it's critical. Therefore, inhibiting its function can significantly impede and hinder bacterial growth. We're going to be with an introduction to classical enzyme kinetics. We're really going to be with some mechanistic stuff. So a little bit of mechanistic kinetics. Then we go on enzyme properties and catalytic mechanisms of enzymes. So the kinetics of enzyme action. Enzymes, like other catalysts, only increase reaction rates by lowering activation energy for reactions that do not affect equilibrium between the reactants and products. Now, there are ways of cheating this system, right? Biology has ways of cheating this system so that an enzyme can drive a reaction that is energetically unfavorable. If it is consuming something else that releases enough energy to make the total process energetically favorable, right? So that one reaction we're really looking at, where we focus on that key molecule, that process is not, the direction the enzyme's taking it is not favorable. However, in the process, say we were hydrolyzing ATP to generate the energy required for the reaction, ATP hydrolysis is favorable. Therefore, in net, hydrolysis of ATP and whatever the step of the biosynthetic process is, the enzyme being catalyzed is in net energetically favorable. Make sense? So coupling these two reactions allows an enzyme to kind of cheat this a little bit. Because in net, it's not, in isolation, yeah, it will not change, so the dogma holds true. An enzyme will not affect equilibrium. It affects the reaction rate and kinetics. However, by coupling reactions, we can cheat that. So if I was to ask you if an enzyme can affect the equilibrium, I'm expecting, unless you give me a specific example where it's coupled reactions so that net equilibrium, but for one synthetic step is against equilibrium, against energetically favorable. I don't want your answer should be that it does not affect equilibrium. Make sense? It affects kinetics, not equilibrium, unless you couple the reactions. And then if you tell me it couples reactions, I want you to give me a specific example. Cool? Reactions may have multiple steps and distinct reaction intermediates, with the rate limiting step being the one with the highest activation energy. That makes sense. You all have seen that before. While the enzyme may transiently be modified in the course of the reaction, upon completion of the reaction, the enzyme is restored to original state. I mentioned that a minute ago. By the way, I apologize for how late they are, but the slides are up on Blackboard. So if you don't have them now, they're up there now. Okay? And we're not going to get through all of them today. We're going to get through a small fraction of them today. Kind of power of enzymes. There is great interest in enzymes because enzymes demonstrate characteristics that we are envious of in our synthetic catalysts. They're highly substrate specific, highly selective in the reactions they catalyze and the products they produce. And they do so under relatively mild conditions. On top of that, their catalytic effectiveness can be incredible. Enzymes can enhance reaction rates by 5 to 17 orders of magnitude. Enzymes are very discriminating in their choice of substrates and products. Where does the energy and specificity come from? One of the answers is rearrange the covalent bonds during the reaction. Another involves non-covalent interactions between the enzyme and the substrate. We're going to get on those. Those more involve our specifics to the mechanism. We're going to get to those. We're talking about the mechanisms, the strategies that enzymes employ. Some examples of enzyme enhancement rates: cyclophilin is 10 to the 5th, carbonic anhydrase 10 to the 7th. We've got the extremes, so urease is 10 to the 14th, succinyl-CoA transferase is 10 to the 13th, and orotidine monophosphate decarboxylase 10 to the 17th. So these are incredibly efficient catalysts. The rate of reaction depends on the relative free energies of the reactants and any stable intermediates in the transition state. The general rule of thumb is that the greater energy difference between the starting material, substrate, and the product, the greater the activation barrier, right? The greater the delta G. Sorry, delta G double dagger for the transition state, right? That's the general dogma. It generally holds true for reactions in general. However, if we can find ways of lowering this activation barrier, this delta G double dagger, to the transition state, we can increase the rate of reaction. A catalyst can increase the rate of reaction by many mechanisms: destabilization of reactants, stabilization of transition state, or altering the reaction mechanism entirely. And the magnitude rate enhancement given by the relative rates of the catalyzed Kcat and the uncatalyzed Kn reactions, with enhancement ranges from 10 to the 6th to 10 to the 14th, reflected in this ratio. Right? It's basically Kcat over Kn. How much increase is this? Excuse me. Comparisons is straightforward for unimolecular reaction. However, with reactions involving 2 or more reactants, the comparison becomes more complicated. Starts becoming 3-body problems. Rate constants for reactions involving 2 or more reactions are second order or higher. However, the enzyme catalyzed KCAT is always first order, meaning that once the substrates are bound to the enzyme, assuming they all bind simultaneously, it becomes a unimolecular reaction, intramolecular reaction. Whereas in solution, if I've got 2 or more reactions coming together, that's a much more complicated kinetic scenario, right? Whereas on the enzyme, everything takes place on the enzyme, and therefore is essentially unimolecular. In enzyme catalyzed reactions, the reaction occurs with the reactants complex of the enzyme. It's essentially a molecular process. Okay. The presence of multiple substrate molecules and/or catalytic groups in an enzyme substrate complex suggests that the catalyzed reactions may be somewhat different from comparable reactions in solution. Such complex reactions in solution become entropically problematic. Such higher order encounters in solution are improbable, meaning that the enzyme brings together a whole suite, a whole environment, an organization of functional groups in catalyzed reaction that is not really conceivable outside the context of the enzyme, right? It's just too complicated, and it's if you combine all those functional groups in solution, you're not going to use the same mechanism. You're not going to have the same interacting groups. The probability is just too low. Interactions of multiple groups in the enzyme substrate catalyzed complex, again, are essentially unimolecular. Everything's bound to the enzyme, and therefore all the chemistry takes place at the enzyme, and then the product's released. We look at enzyme kinetics, enzyme catalyzed reactions. Let me make sure my page now. So enzyme catalyzed reactions proceed more rapidly, primarily because the difference in free energies of the substrate and the transition state is not so great when bound to the enzyme. So it generally is conceived that rather than destabilizing the substrate, the enzymes are working by lowering the energy of this enzyme transition state complex relative to the transition state independent of the enzyme. Right? So that's generally how it's believed these enzymes are increasing the reaction rate. Could be a result of destabilization of the substrate, but that's not really what we're seeing that often. More often you're seeing its formation, the enzyme transition substrate complex is lower energy than the transition state by itself. The enzyme should bind the transition state more tightly than the substrate by a factor corresponding to the increase in reaction rate. Enzymes are not expected to have extremely high affinities for their substrates. If they had a high affinity for their substrate, then you're actually stabilizing the substrate, and rather than lowering this, you're simultaneously lowering this, right? And therefore, you're not decreasing the delta G double dagger as much as you could be if you're binding the substrate too tightly. There's less energy, less motivation to go forward with the reaction, if that makes sense. And so, let's see. This is so that KCAT over KN. The KCAT catalyzed reaction with KN equals the K, the enzyme's transition state complex over the double dagger. This energy for this, the energetics of this. The rates reflect the ratio of the energies of these, the enzyme transition state complex relative to the uncatalyzed reaction. Okay. Binding energy of weak interactions. Energy released upon formation of the enzyme substrate complex is called the binding energy. The binding energy contributes to lowering the activation energy of the catalyzed reaction. Weak interactions between the enzyme and the substrate contribute to substrate specificity and catalysis, and the active site is structurally complementary to substrates, but not completely. We talked about that when we talked about ligand binding, how basically the same interactions are involved with binding, orienting, positioning, and stabilizing bound substrates as we see in other ligands, as we see in protein-protein interactions, we see with proteins folding and structure stability, just to differing degrees. We also talk about structural complementarity. You don't have perfect complementarity. If you had perfect complementarity for the substrate, you're stabilizing the substrate, and therefore you're not getting the same bang you are for stabilizing the transition state. You're not increasing the stability. So if you want a greater preference for binding the transition state, then you want a better energy gain for that than you have the energy gain for binding the substrate, if that makes sense. The active site is structurally complementary to the substrate, but it's not completely complementary. It's more complementary for the transition state. Compensation of the transition state, however, promotes catalysis because it lowers the transition state energy. Energy required to destabilize the substrate is offset by favorable interactions with the transition state, in this case, as you start distorting the substrates to favor the transition state complex. Hosselman proposed that substrate binding that induces a change in the protein conformation in order to induce the active form of the enzyme, induced fit could account for extreme substrate specificity demonstrated by some enzymes. Basically, you have moderate complementarity of the binding site for the substrate, but the binding the ligand to the protein induces changes in the binding site that are only present if it's bound to the right substrate. Does it make sense? They're not really, they're not achieved by binding the wrong substrate. That helps improve specificity. Hexokinase, which catalyzes glucose phosphorylation, provides a good example. You have the lobe structure common to other kinases. The binding of glucose drives the two domains of hexokinase to rotate 12 degrees relative to each other. And as a result, the bound glucose is almost entirely shielded from water. You have a 50-fold increase in affinity for ATP at the same time. It also appears to activate the enzyme for ATP hydrolysis. So binding of the glucose to the glucose binding site it results in a conformational change in the multi-domain protein, which in turn increases affinity for ATP as well as the activity for ATP hydrolysis. ATP binding likely results in additional conformational changes to facilitate the transfer of the phosphorus group from the ATP to the glucose. Right? The induced structural change in the enzyme associated with substrate binding may be required for the enzyme to position the required functional groups appropriately and constraining the transition state and excluding water. So in this conformational chain, you bind the substrate, conformational chain is induced, and in the process, the groups that are interacting with the molecule, the substrate, may change or may distort or twist the substrate in the process. Right? And we'll see that in some examples. I'm trying to get to these. Well, we do see when we start about serine proteases and their mechanism. And we'll start seeing some other groups as well.
Dr. Bishop?
Yeah.
So hexokinase binds at least glucose and ATP
Right.
substrates.
Right.
So is there an order?
Well, this would suggest that the binding of glucose is preferred at first.
Right.
Because your affinity for ATP increases when you after you bind glucose. So it suggests there is an order in this case. That's not simultaneous, and it's not ATP first then glucose.
Maybe if ATP bound first, the affinity for glucose would increase?
It just depends if ATP binding was enough to change. Well, I'm not sure about that one because if it adopted if we favor the shift to the orientation of the domains for binding for the glucose affinity, you're also closing it off, right? So if you induce that, you're closing it off. So I don't know how that necessarily works. I don't know if that necessarily works kinetically, unless you got a really high concentration of glucose, right? So I don't think it works that way.
So the ATP closes off the active
Well,
site.
but in forming the high affinity ATP site, right? In that geometry, we have these domains shifting relative to each other, right? Which closes off the glucose binding site, which is important because you don't want competition for water. So it has to close off the active site in order to eliminate competition for water. Also, all those groups that are now interacting with your glucose, we'll talk about some of these other mechanisms involved. When you close it off, you've now got it in an environment that's under control of the enzyme. So you don't have as much. You have water molecules probably, but you don't have the bulk water. So you're now under an environment that's controlled by the enzyme. You don't have competition as much. It may also result in strengthening and/or weakening of some of the interactions between the substrate and the enzyme. Right? Yes, ma'am.
Can you explain, because I'm having trouble understanding the kinetic reason for the reduction in the activation energy required? So what exactly does conforming the substrate in a transition state do? Like, how does that actually reduce?
Well, so let's say for instance that a substrate has a square. Let's say you've got a so you can distort it multiple ways. And we'll talk about this with serine. We're not going to get to serine proteases today. There's an example of how that happens, where you've got you have a tetrahedral intermediate in the process, and there's hydrogen bond donors, hydrogen bond accepting groups, as well as groups that electrostatically help stabilize charge distribution and distortion in forming the tetrahedral intermediate, right, from a carboxylic acid or from an amide, right? And there's all these groups are positioned so to favor that. So that tetrahedral intermediate is your transition state, or your intermediate, really, right? And you lower the energy of that by all the other groups that are interacting with it that help to stabilize that tetrahedral intermediate. As much as it wants to go back to carboxylic acid or amide, these groups are positioned to donate electron density, donate protons, or pull protons and pull electron densities from other locations to help stabilize that tetrahedral intermediate. Does that make sense? Mm-hmm. If you stabilize that, the energy is coming from the interaction with the protein and not, so that's providing that energy for favoring the reaction, for stabilizing it. So that would lower the transition state energy. Does that make sense?
Yeah.
And there's a few other mechanisms we were talking about too, where the enzyme can dump in protons, pull out protons through acid-base catalysis, and other electrostatic groups, which are a little harder to nail down, to help stabilize the transition state. Make sense?
Yeah.
We go through some, we're not getting through the examples I said today, but when we do, hopefully it makes a little bit more sense of these examples, and if you still have questions about that, we'll get back over it.
Okay.
Is that cool?
Mm-hmm.
You all good with that? Okay. Because I think when we talk about synthetase, it's actually one of the coolest examples, I think, of coupling two reactions to drive a reaction.
Did we talk about that in the slides yet?
We've touched on, I think we've talked about it before. We haven't shown it, and we haven't shown any slides of it yet. It's an enzyme we don't get to. Synthetases are involved with charging tRNAs with the appropriate amino acid. So we're talking about amino acids, we're talking about synthetases. And ligand binding, we mentioned ligand binding because we talked about, I think we talked about editing in there, maybe not, about the selectivity, enhancing selectivity not just through positive, by favoring one ligand, but also through negatives by disagreeing against competing ligands, right? So not only do you increase your affinity, you also decrease the negatives for the wrong one, because you can only get so far with affinity. Well, the enzymes must be able to bind substrates to the transition state. It must be able to release the products. The release of products is often observed to be the rate-limiting step of the enzyme catalysis reactions. And the structural changes of the enzyme that occur in the course of the catalysis result in the state of the enzyme following release of the product to be different from the enzyme at the state of the reaction, start of the reaction. Enzymes use a number of strategies to accelerate reactions. We've got acid-base catalysis, covalent catalysis, metal ion catalysis, electrostatic effects, proximity orientation, as well as preferential binding of the transition state. And that's basically, as I said earlier, that's positioning of the functional groups to preferentially interact with the transition state of the molecule, whether that's in the case of like a hydrolysis, you've got tetrahedral intermediate, or whether it's just a it's a planar intermediate, or something along those lines. It's the groups that are interacting are positioned so as to more favorably interact with the transition state rather than the substrate or the product. Acid-base catalysis, as the name implies, basically involves the enzyme donating or extracting a proton in the course of the reaction. Here we have, as an example, we have the acid-base catalysis donation proton lower energy of reactions with the keto intermediate extraction of proton. Which one is this? This is. Write down. Great. All right. So we have example of acid-base catalysis, right? So here we have a ketone, some substrate up here, substitution up here. We have a keto intermediate going to an enol form. From a keto substrate to enol intermediate, enol product. So that can be result in the loss of this proton kicking up the electrons for a double bond here, extracting a hydrogen to get to here. So there's an intermediate, the transition state here has the hydrogen leaving with a delta plus, just distributes electron density into here, and kicking up more electron density up here in the delta minus, right? And then this ultimately gets extracts a proton. So in our acid catalyzed mechanism, our protein or enzyme is donating a proton. We've got side chains or functional groups positioned within the enzyme so as to put acidic groups in a position to favorably donate a proton to this carbonyl oxygen. Thus stabilizes this intermediate as it donates the proton in, helps to catalyze it. We release this hydrogen that helps us regenerate our enzyme, and that gets us to an enol, right? Alternatively, with the base catalyzed, where instead of dumping a proton here, helping to stabilize this delta minus, we have a basic group here positioned to extract this proton away from the carbene, thus increase the rate at which this kicks back over to form the enol intermediate, right? So in acid catalyzed, you've got groups such as groups that can treat acid-base catalysis. You've got arginine, aspartic acid, cysteine, glutamic acid, histidine, lysine, tyrosine. These all groups have the ability to either donate or accept protons and act as acids or bases and catalyze the reactions. Cool?
Quick question.
Yeah.
So for that base catalyzed, it seems like it'd be pretty unlikely that a base would pull the hydrogen from there. Is that kind of...
Well, this proton is much more acidic than you think it is.
Oh, okay.
Because of the fact that it's got the keto-enol tautomerization here, it's a much more acidic proton than we typically think of as a proton on a carbon, right? So it's much more acidic. So it's not unrealistic to extract that proton off.
Would it also be stabilized by like other residues?
Other functionality can be backbone amides, can be contributing hydrogen bonding to this. You can have you can almost do simultaneously both the acid and the basic steps to further stabilize it, just by the way the end putting in position functional groups, right? So this is more acidic than we were told to think of the carbon-hydrogen bond, and B, you got a lot of other things in there that can help stabilize it. Here's some of the pKa values for some of the common ionizable groups. You have your you get your C1OH plus goes to your ketone or aldehyde. pK is about -7, so that's pretty much not going to happen very often, unless you're talking about as an intermediate process. You position a function ion in such a way that it can dump a proton onto here, and thus help to drive keto-enol tautomerization, like in situations like that. But in general, this direction is not that favorable. Carboxylic acids are glue and acid around 4. Imidazole histidine is about 6 to 7. That's for protonation. Phosphate group 7.2. Lysines 10.5. Arginine. That's an aromatic OH. That's a phenol. Tyrosine is 10.5. The thiol is about 12 to pull that proton in this minus. And then you've got your HPO4 water, and then you're getting to the point where you get your NH2s, pull that proton in, H minus is about pKa 25. So that's realistically, these are not things you'll see that often of immediate pull off of that proton, unless these pKa's here, right, of these various functional groups can be dramatically impacted by other side chains that can be secondary ligands, secondary interactions that help stabilize that. So if I've got a group that's ready to pull the electron density off of this oxygen, right, this proton can be much more acidic. Same thing with the thiol, same thing with the phenol, right? Same thing with the imidazole. You lower those, make them much more acidic and much more nucleophilic by other groups on there. For instance, if I want to make it more acidic, I could put a group here that start actually pulling off this hydrogen. Start interacting with hydrogen. Put something that's called high dielectric, so a lot of electron density next to it to interact with the hydrogen bond with the hydrogen. This makes this oxygen much more nucleophilic. Same thing with your thiols. Same thing with your imidazoles. Cool? And we'll see that when we start talking about some of these enzyme mechanisms. Additionally, the transient formation of covalent bonds between the enzyme and the substrate. It's another mechanism used to drive reaction. And we're going to see examples of this when they talk about both the serine proteases. In fact, it's present almost. I think it's present in all three mechanisms we're going to talk about as far as enzymes go. We're going to talk about carboxypeptidases. We're talking about serine proteases. We're going to talk about the tyrosine synthetase. Are the three examples we're going to go through. We'll talk about examples of enzyme mechanisms. Transient formation of these covalent bonds. As I said, it can form these transient bonds as long as at the end the enzyme is regenerated. So the process, over the course of the process, the enzyme may be changed, but in the end, the enzyme has to be regenerated. Otherwise, it's not a catalytic process. It's not an enzyme. Freely utilizes nucleophilic groups on the enzyme to form bonds with electrophilic centers on the substrate. So it's nucleophilic. Common nucleophiles include the hydroxyls. You got your phenols, less so than your serines and your threonines. Your thiols. The thiol here is one of the most potent nucleophiles, strongest nucleophiles you can have under aqueous conditions. The imidazole is a good nucleophile. We'll see it pop up. This should actually have another proton on here. I said I'm going to draw it, but this should have a proton on it. The imidazole is a great nucleophile. The side chain of lysine is also a great nucleophile. Generally proceed in three. So electrophilic groups you look at, are your carbonyls and your Schiff bases or imines are good electrophiles. They've got this nucleophilic attack right here in the carbon, as well as the carbon here is electron deficient because of the pulling for the oxygen here for the nitrogen. Generally proceed in three phases. The nucleophilic reaction between the enzyme and the substrate leads to withdrawal of electrons from the reaction center. Elimination of the nucleophilic group reconstitutes the enzyme and is one of the last steps in generating the product. So you nucleophilic attack, you activate the molecule for whatever you want to do next, and then in the process you kick out the nucleophile from the enzyme, regenerating the enzyme. Common electrophiles include groups with unfilled orbitals and are bonded to electronegative atoms such as oxygen, such as your carbonyl carbon and your carbon atom and your Schiff base. Metal ion catalysis. Now, in some ways, a metal ion is essentially, it's a positive charge on all pH ranges, right? It's unlike your bases, which when you raise the pH high enough, they'll deprotonate. That metal center has a charge no matter what the pH is. So it kind of, in many ways, is your stable charge regardless of pH. Nearly 1/3 of known enzymes require a bound metal ion for catalytic activity. You basically have two classes of enzymes require metal ions. You have metalloenzymes, which utilize tightly bound metal ions such as iron, copper, zinc, manganese, and cobalt to catalyze reactions. Then you've got the metal activated enzymes that require closely bound metal ions such as sodium, potassium, magnesium, and calcium in order to be active. The metal ion may not be actually actively involved with the enzymatic activity. Right? So one case you have active involvement, the other one involves binding the metal ion for the enzyme to work. The metal ion may be used for anything ranging from binding and proper orientation of substrates to mediate redox reactions through reversible changes in the metal ion oxidation state, as well as electrostatic shielding stabilization of negative charges. So the example we have here is for carbonic anhydrase. We have a zinc coordinated by 3 histidine groups with a bound water molecule. A distant histidine extracts this proton. We have to have a chain of water molecules bridging this. So water is actually actively involved with this enzymatic mechanism, but it's regenerated in the process. So basically, your imidazole and your histidine extracts the proton, which extracts this proton, which extracts this acidic proton on the water that's bound by zinc. We now have our stabilized hydroxide. The stabilized hydroxide attacks the CO2, generates carbonate bound to the metal center. This is displaced by water, giving us our carbonic acid, regenerating our bound hydroxide. Right? And so this enzyme continues. Cool?
So what's this enzyme?
Carbonic anhydrase.
Carbonic anhydrase.
Cool?
Yeah, I felt like I saw it. Felt like
A lot of these are classic enzymes you'll see examples of in your biochemistry lecture. But it depending who you have in your biochemistry lecture, you may have more of a black box approach to studying how they work. If you have more of a biology-oriented class, you're going to have more of a black box. You're not going to focus as much on the mechanism and the chemistry that's involved.
Hmm.
With electrostatic catalysis, electrostatic catalysis, substrates binding often mediated by charge and polar groups positioned in an active site, which is rich in nonpolar side chains. Remember we talked about, if your active site oftentimes has a lot of hydrophobic side chains to help stabilize ligand binding, but then you also have the distribution of polar and charged groups that interact with the substrate very specifically. And in the case of like we talked about with the hexakinase, the conformational shift blocks off from water, and now we're basically, our functional groups are stuck in an environment created by the enzyme. So if you've got a lot of these hydrophobic groups in there, it helps to strengthen those dipolar interactions, electrostatic interactions between the enzyme and the substrate, because you don't have competition from water anymore. Right? We talked about in a low dielectric environment, these electrostatic interactions tend to be tighter and stronger than they are in a high dielectric environment. In a hydrophobic environment is a low dielectric environment. The hydrophobic interactions, however, help provide some of the driving force just for general ligand binding, help provide general driving force for binding. A lot of your specificity, on the other hand, comes from your distribution of your polar and charged groups. Abundance hydrophobic groups side chain affects the pKa of ionizable groups. Direct involvement in catalysis of this electrostatic type of interactions is hard to prove. However, it likely plays a role in drawing in the substrate and stabilizing the transition effect, referred to the Cersei effect. The frequent presence of charged and polar groups in enzyme active sites suggests their involvement in the catalytic process as substrate binding, orientation, and catalysis. Catalysis via proximity orientation. Enzyme catalytic mechanisms resemble those of organic reactions, however, they're far more efficient than would be expected. Two important parameters affecting catalysis are the proximity and orientation of the reaction species. For reaction to occur, reactants must come together in the correct spatial orientation. So if you're randomly in solution, things are bounced together, they have to hit in the right way, and the appropriate functional groups are hitting the right sites to do the chemistry. In the case of an enzyme, when they bind the enzyme is pre-orienting them to potentially react, which in turn significantly increases their effective concentration. Most we talk about the weak interactions and the synergy, cooperativity of protein stability and ligand binding. Same thing is at play here. For reaction to occur, reactants must come together in the correct orientation. An enzyme active site brings substrates, not only brings them together, but also helps to orient them so in order to promote the desired reaction. On top of that, one of the key things, it also binds them and orients them in such a way to prevent other competing reactions, right? So you can have multiple potential, what would, in the absence of an enzyme, would be considered relatively equivalent positions. Because the enzyme binds both these and orients and positions them, it can pick and choose between the different functionalities, similar functionalities on a protein based on the substrate based on its position of the substrate. Does that make sense? It can differentiate between alcohols and a sugar. Right? That's a challenging thing for us to differentiate between individual secondary alcohols and a sugar. But the enzyme can do it because it creates a microreactor inside there that orients the substrates together in order to promote attachment at the desired site. Make sense? That's a huge power that is very complicated. These are big molecules. These enzymes are these big molecules with these little active sites, and a lot of that's going into creating this environment where they can bring the two substrates together in very specific ways, in very specific orientations, to promote the desired reaction and prevent the undesired reactions. So it's not just competition from bulk water, it's competition from potential alternative reactions. For instance, in the alkylation of a ketone, let's say, right? Alkylation of a ketone, reduction of a ketone, or reductive alkylation of a ketone, reduction of a ketone, right? You're going from a ketone to a tertiary alcohol, let's say. or an aldehyde even. Aldehyde, let's say an aldehyde. You get an aldehyde. Aldehyde's planar. The enzyme, through this process, I don't want to do that way. Oh. Where's the eraser? That's what right? These are two different faces of the same molecule, right? Same functionality. The enzyme, because it binds and orients both reactions can differentiate between these two faces, allowing it to control the stereochemistry of the product, right? And that's huge. That's real powerful. What's the name of the drug? Prozac. Anyone ever heard of Prozac? Prozac antidepressant. So back in the 90s, I would say, I don't remember companies, to say Johnson & Johnson, whoever was making it, long, drawn-out synthesis. And they ended up losing all their material because they couldn't do it with great stereo control. And they're getting racemic product. So at some step, there was at least one step, if not more, they relied on a chiral separation to get the enantiomerically pure product. But if you're doing that, you're throwing away all the rest of it, right? This guy E.J. Corey up at Harvard, his group found a way of using a proline amino acid to generate a synthetic. They call it like a chemzyme, whatever you call it. It actually behaved like a little mini enzyme. It took a proline and it's called oxidase of beryllium. I'm trying to remember how I did it. Yeah. Right? It's oxidase of beryllium. All right? So it starts with proline. You get rid of the proline to your proline alcohol through essentially double Grignard. Right? And you put big bulky groups here. What this thing does is actually a mini enzyme. It binds the carbonyl oxygen. Boron has empty p orbitals, right? Right? We have our oxygen here for the ketone, or aldehyde, here. Dumps electron density of the boron. The boron's got orbitals to spare. Small ligand here, because the steric bulk of these things prevents the big things from binding here. The pucker of the proline prevents them from binding the top, so we only bind from the bottom. And so here, it preferentially binds the aldehyde or ketone in such a way that this electrostatic interaction here positions your hydride to attach the carbonyl. And it differentiates the two faces because you can't flip it around. If you flip it around, you don't get effective binding because the big substrate here clashes with these big side chain groups, right? So it binds very specifically. You get a chemoselective ligate, chemoselective reduction, right? This is a huge deal. This allowed him to establish the chirality of an intermediate on the way to synthesizing Prozac from a commercially available product. And they synthesized Prozac, I think, in 4 steps. Needless to say, the pharma company licensed the patents from Harvard, and I'm sure they made a lot of money off of it, right? Because it took a large, long, ornate synthesis and broke it down into 4 steps, and doing so stereoselectively. I mean, and the percent E, they refer to that as percent EE, enantiomeric excess. You know familiar with enantiomeric excess? Right? Enantiomeric excess is just simply how much more of one enantiomer you're making over the other enantiomer you could make. How much excess you make. And this thing is like 99 EE percent. It was pretty wild. But that's just a small taste of how enzymes are working and how enzymes are orienting and putting things in proximity. That whole process of, we talk about the ATP binding, right? The enzyme is here, hexokinase, binds the glucose. The conformational shift, ATP binding. The ATP binding positions the side chains and the phosphates in such a way that they're now in close enough proximity on the right position on the glucose to phosphorate the appropriate hydroxyl group and not one of the other ones. Cool? And this is part of the real power of enzymes. Binding orientation of the substrate, the proper spatial arrangement, and reducing internal motions can promote the reaction and significantly enhance reaction rates, and it results in very specific products. Impact of proximity in experiments involving non-enzymatic hydrolysis of para-bromophenyl acetate. We have para-bromophenyl acetate. The first study of intramolecular reaction, compare an intramolecular reaction in which the bridging segment exerts increasing conformational restriction. So here we basically have, we're starting off with essentially an intermolecular reaction. You have your acid, it reacts with the acetate here to form the anhydride and give us back our bromophenoxide, right? But let's do it intramolecularly. Now we have a 3-carbon chain, right? 3-carbon chain reacts with it to form the this 6-membered ring anhydride, right? Alternatively, we have a 2-carbon chain here connecting it, same reaction, 10 to the third times faster, 10 to the 5 times faster. But if I had a 4-carbon chain, this reaction doesn't happen. If I have a 7, 8, or 9-carbon chain, it's going to be much slower than it is with these 5 and 6-membered chains. And if I get beyond that, larger than that 9-membered chain, say 10, 11, 12, beyond that, your kinetics are going to start approaching what you have here with intermolecular, right? Remember your organic chemistry. The 6-membered ring, 5-membered ring transition states and intermediates are kind of a magic combination as far as accommodating carbon's preferred geometries, right? You remember this?
Yeah, it gets strained if
you get 4 strained, 3 is really strained, but then 7, 8, and 9, the geometries aren't quite working. They're less favorable because the ring's too wide, and then once you get to 10, now you've got so much conformational flexibility, you're starting to approach intermolecular kinetics versus intramolecular kinetics, right? 7, 8, and 9 aren't very good. That's why there's a drug called Taxol that had an 8-membered ring. There's a big drug target, high cancer drug that was a huge deal they finally synthesized in the laboratory because they're trying to figure out ways of making enough of it as a drug. Eventually, they figured out actually they find better ways of extracting it from yew tree leaves. But all right, so that's simply just basically what can happen if you position things close enough together and put in the right geometry. So in enzyme catalyzed reactions, the reaction occurs with the reactants complex of the enzyme and essentially intermolecular mechanism. We talked about that. The greater the number of reactants involved, the greater the advantage provided by the enzyme. High effective concentration and substrate concentration in the active site, associated orientation, in the active site. At first glance, this would not appear to be a factor for unimolecular reactions. However, virtually every chemical reaction can be catalyzed in solution, and the observed rate of reaction depends on the concentration of the catalyst. Nucleophiles, electrophiles, acid-base catalysis, etc. So even in the case of intramolecular reactions, the binding of the enzyme can significantly increase the rate of reaction. Enzymes possess numerous functional groups in the active site that could potentially play roles in nucleophilic acid and base catalysis, we discussed, results in very high effective concentrations relative to the substrates. Relative to uncatalyzed reactions, acid, base, and covalent catalytic mechanisms can be estimated to increase reaction rates by 1 or 2 orders of magnitude. Orientation and proximity can get you 10 to the 8th fold. So between the two of these, you're getting about 10 to the 9th, 10 to the 10th fold, right? But sometimes, some enzymes achieve 10 to the 14th, 10 to the 17th fold, increasing rates of reaction. So you're exceeding what you can simply explain by this orientation, proximity, and simple acid-base catalysis. You've got more stuff going on. That brings us back to transition state binding. So all this other stuff, one of the most important catalytic mechanisms employed by enzymes is preferential binding and stabilization of the reaction transition state. So in addition to bringing them together, you also introduce interactions that promote them to react when they're together, right? To help stabilize the intermediate. Enzymes lead to strain, distort substrates into transition state geometries through binding sites that optimally bind the transition state. You achieve these distortions that are energetically costly because you're paying for that through the interactions between the enzyme and the substrate. Does that make sense? You get twists and distortions of bonds, distribution of electrons based on the interaction with the molecule, the protein, right? Just by this mechanism alone, through the stabilizing transition state, you get a 10 to the 6-fold increase in the reaction. So by the mechanism, a 10 to 6-fold increase will require 10 to 6-fold enhancement in transition state binding relative to substrate. 8.1 to kcals per mole at room temperature. It's a very large rate enhancement would be achieved by the formation of a few new bonds interacting between the transition state and the enzyme. A few hydrogen bonds, a few dipole-dipole interactions, a few electrostatic interactions would be sufficient to get this 10 to 6-fold increase in stabilization of the transition state. So the transition state binding may also contribute to enzyme specificity. While molecules similar to the substrate may be bound by the enzyme, they're incapable of achieving the transition state and therefore no reaction can occur. This is supported by examples where you've got transition state homologs or mimics being used as competitive inhibitors for the enzyme because they bind tightly to the enzyme, more tightly than, say, the transition state. However, they're incapable of going forward in forming product. One example is this dihydroxyacetone phosphate being converted to glyceraldehyde 3-phosphate. You have an enediol intermediate. If you do 2-phosphoglycolate, which roughly replicates this geometry, you got partial negative charge here. You have partial negative charge here. You have the phosphate groups here. Just the position of these functionalities here is such that it replicates some of the electrostatics of the enediol intermediate, and therefore it is found to bind to the enzyme active site competitively with the enediol intermediate, and therefore inhibits conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Reaction involves general acid-base catalysis, formation of enediol intermediates. 2-phosphoglycolate reproduces the partial charge, this delta negative and this delta plus down here. We have delta negative down here, delta negative up here, and then full negative here. So it replicates is this portion of the charge. And that is where we're going to end today. So, any more